tell you what I'm going to be telling you about, so you know where I'm going with this. I'm going to give you a little bit of background on the system, and background on my pathway. Then I want to go into my two main research parts that I want to talk with you today about. And the first is about a regulator of TGF-beta signaling, and the second is results from a screening that I performed to identify additional regulators of the signaling pathway. What is transforming growth factor beta? It is a superfamily of molecules. There are over 30 that are needed in us. Every eukaryotic system that has more than 2 cells needs the cells to talk to each other. And this signaling family is one of the many ways that these cells talk to each other. So one cell will secrete this molecule. I represent it here as 2 little circles because it acts as a dimer. This dimer then is received by usually a receiving cell at a receptor. These receptors then become activated by a phosphorylation event, which then in turn activates molecules that can go into the nucleus and regulate the transcription of target genes. Am I good? Actually, this means good and it also, okay. I'm good. Okay. And so what this little TGF-beta, transforming growth factor beta, is a small peptide, a pretty tight little knot. It's called a cysteine knot because there are disulfide cysteine bonds in it. Okay. And so next, I want to tell you, go back from the molecular signaling part of it into why this is important. What are the effects of this family of signaling pathway? And so 2 little signaling by a TGF-beta can lead to, this is a muscle-specific one called myostatin. It's also known as GDF8, growth and differentiation factor 8 in us. This was a natural variant that was found. Usually this TGF-beta puts the brakes on cell growth. When those brakes are removed, then you get muscle hypertrophy, overgrowth. And you can see this Belgian blue cow is really muscular without even trying. It's been identified in humans. The homozygous loss of function allele in this growth and differentiation factor 8 leads to a muscular baby who's probably grown up a lot by now. Any of you have had babies or been around babies know that this is not normal baby fat. He was the son of 2 athletes. However, on the other hand, if there's a gain of function mutation, that can lead to muscle wasting, which is found in a form of muscular dystrophy. Another one is involved in bone growth. And so this particular one can lead to a very common dwarfism syndrome that also involves polydactyly. Too much signaling by a TGF-beta, a mutation in a negative regulator of this pathway. When you take away the negative regulator, you allow more signaling to occur. And in this case, it leads to overgrowth. These 2 girls are twins. She has Marfan syndrome, and even at 8 years old, you can see that she's already a head above her sister. So I actually bring in the sister to talk about this in my genetics class. And so you can see with its effects on all types of cell growth, bone growth. Oh, what do you know? One of our own is working on this. It's also involved in speciation, and not just the ordinary differences that occur between you, me, and your neighbor. Darwin's finches, one of the most famous things about speciation, examples is Darwin's finches and their beaks and what they use. Differences in signaling by one of these TGF superfamily members, bone morphogenetic protein 4, is correlated with changes in the beak size. More of it leads to more bone growth, or beak growth. And with BMP2, a similar thing in mouse. There's less BMP2 in the developing digits than in the bat, which has more. And you can see there's an extreme difference in the length of these digits compared to a mouse. And also compared to the hind limb. So I like to think of molecular pathways in a developmental sense. And because I have that bias, I think of cancer as just a developmental process that's gone wrong. Because a lot of the things, cancers will hijack developmental processes to survive. And so, our TGF-beta is also involved in cancer. You saw the roles that it has in development. It also plays roles in homeostasis. And when those processes go wrong, that's when the cancers can occur. And so, with the normal roles, like you saw with the Belgian blue cow, when it goes wrong, then you can have a cancer. The superfamily is involved in morphogenesis. Basically, it's cell-to-cell communication. It's a population, just like humans. We're constantly talking to each other, and cells are saying a lot of the same things that we say to each other. I have 2 little kids. Now they're 9 and 7, but I'm telling you, grow up. Do something. Don't just sit there. You got to get ready for school. You got to do this. You got to do this. You got to go there. And the same thing with cells. They're constantly talking to each other and telling each other where they are at and what their neighbors need to be doing. And so, also a little help, please. Or even go away. As a mom, I hear that. Again, the TGF-beta superfamily is also involved in the cell migration, in the remodeling of the matrix around these cells that can allow a cell that's normally bound to actually migrate and invade and go somewhere where it usually doesn't go. And so, when they're hijacked in cancer, this can lead to uncontrolled tumor growth. These are all things that define a cancer cell. What are things that define cancer? She's actually taking a picture of this. That's awesome. Okay. They grow when they're not supposed to grow. They divide when they're not supposed to divide. They survive when they're supposed to die, right? And the other thing that tumors do is they need a source of nutrition. And so, and they need to be able to they can metastasize, they can go away from their normal environment. And all of these can be caused by hijacking the normal roles of the superfamily. Any questions? Okay. So, you can see that differences in the signaling levels of the superfamily are critical to the output, to the phenotype, to the morphology, the physiology of the organism, of the individual. And so, how is this regulated? By finding out how it's regulated, then maybe we can re-regulate it when it's dysregulated, like in cancer, like in the developmental disorders. And so that's where my work comes in. And so I want to give you a little bit of background on that. So at this level of the secreting cell, it's initially, you can see that there's a level of regulation that is known that occurs between the secreting cell and the receiving cell. There's an intercellular matrix, or also called extracellular matrix, that can keep this TGF-beta, here shown in white, 2 balls, from interacting with the receptors. There are a variety of other things that are known. Not all of them are known down to the molecular level of the molecular interaction. But what I'm interested in is identifying novel regulators and defining how these regulators interact with the signaling pathway, either at the level of the signal itself or at the level of the receptor. This part is actually fairly well fleshed out in model systems, in cell lines, in cancers, but the extracellular has been harder to get out. And so that's why I'm using a model system. So I mentioned that TGF-betas are highly conserved, right? Eukaryotes, anything more than 2 cells, you conserve down even to sponges. Okay? In us, we have over 30 of them that are needed. They're subdivided into these groups. You've seen these groups before in the introduction, the bone morphogenetic proteins, the growth and differentiation factors, TGF-beta and the Marfan syndrome. It's complicated. It gets really complicated and messy in us. Sometimes, if you don't have enough of just one, it can be lethal. You can also have a great developmental disorder if you lose just one. On the other hand, there can be some redundancies in the functions, such that if you knock down the function of one of these members, another one will compensate and you'll never actually see a defect. And so we want to study this system, this family, in a simpler system. And that's where the power of the model systems comes in. For instance, in flies, there are 8 of those, a lot simpler. Still a few overlapping phenotypes. And in worms, nematodes, actually, not actually worms like annelids, like earthworms, like bait, but nematodes. There are only 5. And actually, 2 of them just show sequence homology to the superfamily, but they haven't had a role ascribed to them or identified yet. That leaves only 3, not the 30 plus that are in us, but only 3 that actually play a physiological role in the animal that we can tell. And what's more, when you knock out any one of these 3, the worm survives. It's fine. It's viable, which means it's a lot easier to work with. It's not easy working with a lethal phenotype. The second thing is that they have non-redundant phenotypes, non-overlapping phenotypes, which also makes it a lot cleaner to work with. Okay, any questions on TGF-beta signaling at this point? Undergrads? Grads? Postdocs? Either I'm being very good or I've lost everybody already. You're just like to tell me. Okay, let me tell you about my system and the power that C. elegans can bring to this problem of identifying and characterizing the regulators for TGF-beta signaling. You can also take this and apply it to whatever your favorite question is and see, with C. elegans, my nematode model system, can that be applied to other questions? I'm going to give you some of the foundations for this system right now. How many of you have studied, know a little bit about C. elegans? Okay. Okay, good. Good. I think I've prepared it well enough. Okay. C. elegans is not this big. C. elegans, when fully grown, is only 1 millimeter. 1 millimeter. You can grow a ton of them in a small amount of space. Biochemistry and growing up vats of them makes it problematic, but for genetics, it's beautiful. Very easy to manipulate, very cheap and expensive, easy. A dissecting scope manipulations. It's a simple multicellular organism. Every single one of its cells is the same from animal to animal unless you've got a mutant, which is very much unlike us. 959 somatic cells in the hermaphrodite. Every one of them has a name. Only 1,021 for the male. You do the math. There's the difference in, and it's a hermaphrodite. Oh, good. Hermaphrodites. Which means what does it mean to be a hermaphrodite? Somebody from the third row. Hermaphrodite. It makes both sperm and it makes oocytes. It self-fertilizes. It doesn't need sexual intercourse. It means you can put one hermaphrodite on a plate. And in a generation, you've got 250 F1s. When you're doing genetics, that gives you some really good statistics. But there are also males. And so when you're doing genetics, you want to make new genetic crosses, you want to make new genetic backgrounds, make new strains. You can do that too. It's beautiful. Okay, and you can do it quickly. In a week, you can have grandkids. Not you, your mom. So, which also makes it relatively fast. It's not yeast, but it's not mouse, it's not corn. And we don't really care about C. elegans, nematodes. They're non-pathogenic. They aren't, other nematodes are a major pest, but they also aren't us. And we're navel gazers. And I also work at a health science center at a medical school. And so what's really important is that things that we find in C. elegans can be applied to higher systems. Their genome is highly conserved, and the genome was the first multicellular animals that was completely sequenced. So we can mine that data. But it has fewer genes. It has about 20,000 genes. Again, 30 plus TGF-betas in us versus only 5 in C. elegans makes it simpler. We can knock down gene function either by mutagenizing the DNA or by knocking down the translation of the protein by taking out the RNA using a technique called RNA interference. Show of hands, who knows RNAi? Okay. You're able to remove the gene product, as effectively a protein, by doing this technique. What's even cooler is that we can add genes back into this organism really easily. We just use a little tiny needle and inject them, the gonads. Okay, and so what I do in my lab is I use a combination of genetics and biochemistry, molecular biology, cell biology, and imaging. And you're going to see all of that in this lecture today, in this seminar. So I'm going to go back because I want you to have at least a working knowledge of some of the jargon that I have to use when I'm talking about this. My TGF-beta is called DBL1 or double 1. It's named after the BMP and the fly DPP. So DPP and BMP like 1. Okay. And mine is involved in normal body size. Remember, you've got the dwarfism, you've got the Marfan syndrome, overgrowth syndromes. Well, in worms and our nematodes, it also affects body size. This is a negative regulator of signaling, and so if you remove the negative regulator, what will happen to the worms? They become long. If you remove the core signaling pathway, then the animals become small. And that's the main phenotype that our lab uses. Nice, beautiful dissecting scope phenotype. So here's a normal animal, really, again, only 1 millimeter, not 3 feet, with reduced signaling. How many of you can see that phenotype? Yeah, okay, good, thank you. And then with increased signaling, the animals become really long. It's so cool. It's additive, too. And so you can get, we have the longest worms in the world in my lab with a triple mutant.
How long?
About 150, about 50% longer than normal. And so, right, where's the viability? The viability, I won't get into that now. Okay. But, okay. We have clones. Not only do we have these body size changes, but they're also very quantitative, and they're very tight. You can see by this is so the variation from worm to worm, from animal to animal, is very small. And so what you can see here, standardizing to the normal animal, you lose the function of RTGF-beta. The animals are about 80% the length of wild type. You add back using a transgenic technique. You add back more of that, and the animals become longer, in part because the receptors are in excess. You take away that negative regulator. Again, it's in excess. You take away the negative regulator, and you add even more of the TGF-beta, and the worms become freaky long. Okay. So now a little bit of cell biology. This is the worm. It's got a lot of the things that we have. It's got the intestinal tract. It's got a reproductive tract. It has muscles. It has nerves. It also has epidermis, and that's represented in the tan here. Just a very few number of cells makes up the whole epidermis in this animal. And the reason that I'm bringing that up is that the hypodermis is the receiving cell for this TGF-beta, or DBL1. If we take a cross-section of the animal and eviscerate it, so take away the intestine, take away the gonad, you can see the epidermis here. And I want to focus your attention on just this part. There's a ventral nerve cord that runs the length of the animal like that. And that is actually where our TGF-beta is being made. It's being sent, and then the receiving cell is right next door to it. It doesn't have far to go. And so people will say to me, this is a signaling system. It ends up in the nucleus, regulating the transcription of a wide variety of genes. And so in my previous lab, we did a microarray analysis to determine what genes were highly regulated, either upregulated or downregulated. And you got a lot of what you might expect. If you're a long animal, there's an increase of genes involved in metabolism, in protein and nucleotide expression, and also in structure. So extracellular matrix. The hypodermis secretes a cuticle. It's like bone, but it's flexible. It makes the animal resistant to a lot of environmental insults and, helps it survive. But there's also a chicken and egg thing. Are the animals long because they express all these, they regulate all these genes, or do they regulate all these genes because they're long? So I can't answer that right now. But what I can do is I can show you what my grad student found, in that normal cuticle looks like this. It's got nice layers differentiated in that cuticle that protects the animal. And if you have less than normal TGF-beta, this layer right here is looks more filamentous, it looks more screwed up, and here it looks denser, and here it looks looser, more expanded. And so we're thinking it also, this layer here is also rougher than, or less organized than what we think. And so we're thinking that cuticle plays a large role in body size. But these animals are not just, it's not just like a squished cuticle. There are cuticle collagen mutants that create a dumpy phenotype, which is distinct from the small phenotype. So there's more to it than that. Okay, but now let's get back to signaling.